without further delay, let's continue because we have another LS fighter today with us, which is our next speaker, Bernie Zipprich. Bernie is also an LS patient. He is an accomplished healthcare innovator, strategist, and entrepreneur. He was diagnosed with sporadic ALS in 2020 at age 32. He founded Project Mosaic, a program aiming at bringing a new class of innovative ALS disease models and an AI-based platform for conducting phase 0 technical trials in a dish to market. Bernie and his team are partnering with researchers, industry partners, including iXCells, and ALS nonprofits, including ALS Network, during this initiative. Beyond Project Mosaic, Bernie is also a NEOS certified ALS research ambassador, a member of the ALS Association Board of Trustees, and is a recognized thought leader on ALS scientific strategy. Bernie lives in New York with his wife, Marie Zipprich, and their young son. We are honored to have him and his wife joining us today, and as well as his Project Mosaic team members, Eugene and Tusha. So because of Bernie's condition, Eugene Brandon is going to be present the work and the mission about their initiatives from lab to market, decoding the LS mosaic to enable precision medicine at scale. Eugene. Oh, really? Oh, sorry. Sure.
Hello. I just want to say that it is such an honor to be here, and I've so loved hearing all of the other presentations today, and maybe as a way of introducing what we are hoping to do with Project Mosaic. One thing that is incredibly exciting about ASOs and the precision medicine techniques that we heard about before is that they allow us to target rare diseases at the source, and as many of you probably know, and as we will discuss in a moment, one of the big challenges with not only ALS but other neurodegenerative disorders like Alzheimer's, Parkinson's, and FTD is that many cases don't have an obvious genetic target to treatment. So a big thing that we are trying to do is develop ways of being able to identify both targets and promising drugs that that might work for subsets of of patients with these diseases. And then the last thing I'd like to say before I hand it off to Eugene is that is that that one thing that I love about working in this field is 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 mission driven and 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 collaborative. So so many people are and and I and I am and I'm so excited that we will also be hearing from 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 Dr. Don Cleveland and 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 Dr. Justin Cheetah because because a lot of the of the science that we are now now trying to build on and scale we 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 are a a big a big a big the, the gratitude to to their research as well. Using.
Thank you. Thank you, Bernie. I want to thank iXCells for inviting Project Mosaic to present today and for having a session on ALS. As Bernie was just pointing out, it's pretty interesting that the rarest diseases, the gene is often known, and then you have to address it in that way. And here we're almost coming in as one of the least rare diseases, meaning there's a prevalence of 30,000 in the U.S., but an incidence that's much higher, equivalent to multiple sclerosis, for example. And in the vast majority of cases, we don't know what the gene is, or if there even was a gene that was involved in the source of the ALS incidence or progression. So here's the team for this is a early stage in this operation, the Project Mosaic, but we're launching fast, as you'll see in the presentation, and want to acknowledge Bernie for all the work he's done to get this to where it is today. I first met Bernie a few years ago when he joined the board of the ALS Association, and he started telling me about this idea of neurobiopsies for patients. And at the time I was a little skeptical that this could be something that would be sufficiently effective and sufficiently cost effective to really attract investigators and investors. But now things have changed. There's the technologies have evolved, and the landscape is different, and there's just things are accelerating. It feels like the whole field of ALS has been accelerating over the last few years, and I think now is the time that a Project Mosaic is actually really needed. So we'll talk about that right now. So because these are Bernie's slides, and I just joined the group, I'm going to read my talk, which I admitted to Don, it's a little bit embarrassing. It's a little unorthodox for scientists to read a talk, but I think that's what I need to do this justice. So what is the vision? What is the motivation? And neurodegenerative diseases are tragic, and everyone is different. Everyone is different. And that's kind of the recurring theme here. What if we could cure devastating diseases like Alzheimer's, Parkinson's, or ALS with treatments tailored to each patient's unique biology, unlocking a future where precision medicine is what transforms lives. So this is obviously a highly ambitious goal, yet we see a path which firstly involves creating an in vitro avatar for each person, which we'll talk about now. We're extremely excited to be embarking on this journey. So where are we today? Let's consider the current state of affairs for neurodegenerative disorders. There are millions of people. This is not rare to have a neurodegenerative disorder. If you consider Alzheimer's with nearly 7 million people in the US, Parkinson's over 1 million, and the other neurodegenerative disorders combined, this is 10 million people in the United States, costing approximately, by some estimate, $650 billion. And this is largely carried by the Medicare, Medicaid, and insurance, but also importantly, families. This is one thing that we come across a lot with the ALS Association, is that families are really taxed financially dealing with their disease. These neurodegenerative diseases, adult onset neurodegenerative diseases, are for the most part sporadic, with no particular genetic cause or even environmental cause identified. So this has been plaguing the field, in particular ALS, for forever. And it's still something that we think we can start to ferret out, as research continues to unfold. Tragically, there are no effective drugs to slow the progression of these diseases for sporadic ALS. So Don will talk a little bit in a minute, I'm sure, about the antisense oligonucleotide for SOD1 ALS. And on top of that, there's a high failure rate. So the rate of drugs not making it to market for neurodegenerative diseases is 10 times greater than for other CNS disorders. So this is really a situation that's screaming for a new approach. So let's consider why drugs are failing. Firstly, there are numerous potential causes of disease, and in many cases, there may be several causes interacting to initiate and propagate the condition. Thus, testing a drug addressing one specific molecular target may be insufficient. Even identifying a target may be difficult. And importantly, the course of disease and the nature of disease can be very different between patients. This heterogeneity is seen in all of the neurodegenerative disorders, but ALS is particularly notorious for it. Typically, it's said that the prognosis is 3 to 5 years with ALS, but from what I have observed with my ALS friends and colleagues, admittedly, this is anecdotal, there seems to be at least as many exceptions to this prognosis, with some pals dying in less than a year from symptom onset and others living a decade or more. Clearly, a one-size-fits-all drug development program is not working for neurodegenerative diseases, and given the heterogeneity, maybe we shouldn't even expect it to work. Despite the heterogeneity with neurodegenerative conditions, research has revealed a common feature across these diseases, and especially prevalent in ALS, which is loss of function of a protein known as TDP-43. TDP-43 is an RNA handling and processing protein, which generally operates in the nucleus of the cell, but in many cases, including over 95% of cases of ALS, TDP-43 protein is found mislocalized to the cytoplasm. And in the cytoplasm, it is prone to aggregation, becoming insoluble and dysfunctional. So as we consider the platform we are building at Project Mosaic, we will pay special attention to TDP-43 loss of function as an important feature. So what is Project Mosaic? Project Mosaic aims to transform the way we view neurodegenerative disorders and revolutionize treatment development, driving more precise and effective therapies. What do we mean by this? Well, let's consider cancer by analogy. 100 years ago, cancer was considered as a single disease which can affect different organs and systems, but which was really best treated with blunt tools such as radiation and surgery, with obviously limited efficacy. Nowadays, we know so much more. For example, within breast cancer alone, there are at least 4 different forms, which can have very different prognoses and susceptibility to treatments. Thus, you will not be surprised that in cancer research, drug screening can include testing on hundreds of different patient cell lines at a time. The PRISM lab at the Broad Institute is a great example of the kind of initiative and represents an effort which Project Mosaic may seek to emulate. The PRISM lab has over 900 cell lines from different patients and cancer covering different cancer types, with extensive clinical and biological information associated with the lines. Hundreds of collaborators are partnering with PRISM, including over 30 biotech and pharmaceutical companies, reflecting the incredible opportunity to gain critical knowledge using such a powerful system. Project Mosaic aims to do the same for neurodegenerative disease. So why ALS? Obviously, Bernie and I and others have a personal interest in ALS, but at the same time, it represents a great starting point for this approach. That is, a great deal of scientific effort has been invested in ALS, and we already have industry-ready sporadic disease models and commercially ready iPSC cell lines with ex vivo data. As well, ALS pathology is concentrated in known distinct cell types, and the potential for precision medicine to be effective has already been demonstrated with the recently approved ASO to SOD1 known as tofersen or Qalsody. Importantly, recent advances in iPSC-based models suggest that a personalized ex vivo approach is possible. Scientific reports from the Achita lab at USC and the Coyne lab at Johns Hopkins have shown disease phenotypes in vitro, including TDP43 loss of function and the potential for therapeutic intervention. Yet some barriers to adoption remain. These include demonstration of reproducibility and reliability in the industry setting, questions as to the volume of data that will be needed for regulatory acceptance, and correlations to patient disease characteristics, including responsiveness to approved therapeutics or therapeutic candidates. And reports have shown that, like the heterogeneity of the disease itself, there is heterogeneity in how well ALS patients' cells respond to drugs in vitro. These data suggest that there is an opportunity to stratify patients in other ways than just the stage of their disease or the course of their disease, as we seek to tease the subtypes of ALS apart and improve clinical trials. Project Mosaic seeks to move the approach from academic research to commercial utility. And in November 2024, Project Mosaic had a meeting of the minds to explore the possibilities, consider what issues might need to be addressed, and make a plan to get the project rolling most efficiently and effectively. You can see there, there was both drug makers, thought leaders, academic labs, government and nonprofit groups, as well as CROs, including iXCells at this meeting. And here you can see Project Mosaic was fortunate enough to attract some of the best in ALS from academics to industry for this meeting in Washington, D.C. last November. So the moonshot outcome of the meeting is a goal to make standardized sporadic ALS disease models and a PRISM-style AI drug testing platform, and to have these available commercially within the next 3 years. We don't consider this just an aspirational goal. we believe that with sufficient resourcing, it can be achieved. So how are we approaching this ambitious goal on a practical level? We need to start somewhere. So we take the crawl, walk, run style ramp up. Learning to crawl in stage 1 involves getting the cells up and running from a relatively small number of donors and establishing the endpoints and the protocols for this foundation of the project. This will culminate when the systems are internally validated and reliable and reproducible with a track record of success. Stage 2 will involve so-called external validation, establishing algorithms to mine the data and assessing the connections between results in vitro and patients' characteristics. Stage 3 will be the scaled clinical trial in a dish, whereby we have a system of sporadic ALS in vitro, which can be used commercially for research on ALS, for drug screening and drug development. Ultimately, we would like to have a system which can be used to predict which course of treatment is going to be most effective for a given patient. We are grateful to iXCells for partnering with us on this. As well, we have fantastic tissue and data partners lined up in Answer ALS, ALS TDI, and Genius, the data company. So we are on track for beginning this work now. And I'm very excited for Neon Way, and we'll be having a meeting tomorrow to keep this rolling. Indeed, we can't afford to wait. The Project Mosaic system is needed as soon as we can get it up and running. And there are clinical trials in ALS today which could benefit from Project Mosaic's inclusion. So, Bernie, let's finally make neurodegenerative disease treatable. Are you in? Thank you.
Thank you, Eugene. Thank you, Bernie.